today. We're going to go ahead and start with Dr. Austin. Just want to go to the next slide. And I believe the link to the slides is in the chat. So this is our agenda for today. Our goal is to really give an overview of the various facets of the N3C project, and also just introduce the community to those folks who are leading these various components, so that you can start to get to know who people are and what different people are doing in this context. There are many, many other people that have contributed significantly to the N3C, and so we're hoping we can give you a little taste of some of those thought leaders here. So we'll go on to Dr. Austin.
Okay, thanks. Boy, that's a really packed agenda, so I'm going to run through these really quickly. My role today is just to welcome you all and thank you for all the incredible work that all, and I mean all of you, have done to get us to this point, and we will need all of you, again, all. Those of you who've been deeply involved in this so far, and those who may not have been deeply involved in this so far. We're not going to get where we need to go without everybody on the bus, or perhaps we need something even bigger, a boat for this one. The data that we need, the diversity of data and approaches and expertise that we need is going to require the best in all of us, and I really appreciate you're all contributing to this, and those of you who are relatively new, welcome. I just want to let you know that yesterday we were able to go live with the N3C site on our on the NCATS page, so there's a citable object there now. We've been asked for that, you will be glad to know, by a number of other institutes at the NIH who are interested in having their investigators and networks contribute data, which is exceedingly good news. We also have a general COVID-19 translational science page that you might want to take a look at. Next one. So what is the point of this? Though there has been a lot of work over the last really 3 years by all of the CTSA hubs, the informatics people at all the CTSA hubs, CD2H, the NCATS informatics staff, many, many other people. This has really been brought to the demonstration phase in the context of COVID-19. And of course, the word, buzzword of the day that we are all living with is the word rapid. We always think at NCATS about getting more interventions to more people more quickly, but this has really upped the ante. And we're talking about the rapid collection of data, clinical lab diagnostic data from hospitals, healthcare plans, the organizations that in which you all work, and to do it as close as we can to the peak of the epidemic. In some states, unfortunately not in Maryland, where I am speaking to you from, we're still peaked, but in other places this is decreasing, and we want to capture those data as the pandemic evolves. And I think we're going to see a number of system sequelae of this disorder, which are going to affect us and our healthcare systems and the health of our patients for some time to come. So of course, speed is critical. We have to make access to the data fast and easy, that we don't tell people what the analysis would be. They get to decide what it is and do that in an iterative way. And we want to be able to make these data, make the platform usable for the more modern methods such as ML, AI, those kinds of things. And eventually to evolve to support clinical trials. Next one. So last thing I just want to say is that there is a little bit of funding available to support data transfer to the hubs who need it. The hubs that are able to contribute data immediately, you are able to contribute data immediately, and many of you have already. And we hope most of you will be willing to do that with the funds that you already have from us. However, we do have some limited scholarship funds are available to support personnel and resources for data transfer. And I'd ask you to think about that in the typical way that we think about scholarships. If you really need it, it's there. But if you don't really need it, please don't ask for it, and it will be available to those who really do. So if you do, then you have to apply to this emergency NOCI. We've got a bunch of other NOCIs out there, but the one you want is 028. It has to come under this for arcane funding reasons. So please use that one. And you can see the numbers down there. And if you have any questions, of course, just contact your program officer or Claire or Mike or Joni or me. We will get you taken care of. So again, thank you all for your efforts. We are just at the beginning of this. So we got a lot of work to do, and I am really excited about the real transformation that this represents for our community. This is huge, and I want to thank you all for getting us here and what you will do to get us where we need to go. So thank you. And that's all for me.
Wonderful. Thank you so much, Dr. Austin. So I'm going to give a very brief overview and then we'll continue along the tour. So I'd like to introduce what the National COVID Cohort Collaborative, or N3C, actually is. As Dr. Austin mentioned, it's really, a centralized, secure portal for hosting patient-level COVID-19 clinical data and deploying and evaluating methods and tools for clinicians, researchers, and healthcare. And I wanted to mention that, unlike many other kind of smaller data sharing initiatives that require sharing the data in order to have access to the data, our goal with the N3C is to make this data as widely available as possible to experts in many different areas of analytical science. So really bringing our nation's best experts in machine learning, as well as in clinical care and all kinds of different translational sciences together to work on the data together. And so because of the security afforded by us in realizing the N3C, we are actually legally and ethically allowed to support a much wider diversity of collaborators in this context. I wanted to mention some key partners. This is really, a program that is led by the CTSA program in collaboration, and I'll talk a little bit more about this, with the Distributed Clinical Data Networks. That's PCORNet, Odyssey, ACT, and Trinetics have been wonderful partners. You'll hear more about that from Emily, as well as many other clinical partners and collaborators. And while the CTSA program and NCATS for the funding has been responsible for launching this program, it is by no means limited in any way to only those investigators that are funded by NCATS or are part of a CTSA. It is being rapidly organized into 5 work streams, and you'll hear from all of these work streams as we go through: data partnership and governance, phenotype and data acquisition, data ingestion and harmonization, collaborative analytics, and synthetic data. This was sort of a self-organizing process, and so this is kind of where we've ended up. It's a little bit dynamic, so bear with us and give us your feedback as we go along. Okay, the next slide, please. So I wanted to sort of dispel some maybe concerns or misinformation about what is different about what we're trying to do here from some of the existing data sharing efforts. And so I've had the question quite a lot, but am I not already sending data? So many of the clinical sites are being asked quite a lot to put together different kinds of data for COVID, and are participating in federated querying networks such as PCORNet or Odyssey, as I mentioned earlier. What's actually happening in those contexts is not really data sharing, but query results sharing, if you will. And so this is indicated on the left side, where a user might ask questions such as, how is time on the ventilator impacted by a given drug? And that question then would be sent to the network data partners that are part of that network, whether it be ACT or PCORNet or Odyssey or Trinetics in this case. And then the results of those queries would be sent back, and you could do summary analytics on those results. And so that is the sort of process by which a federated querying network functions. And they're incredibly valuable and have been incredibly valuable in the context of COVID for getting things rapidly stood up so we could start asking those kinds of really important questions. They're also because of that enormously difficult work that's been going on for years, both in the context of the networks as well as in each institution that has pushed their data into one or more of these different models, we are actually able to instantiate the N3C only because of that work and only in partnership with those organizations. And because of that, we can now start to do what is over on the right, which is centralized analytics, where we can ask questions such as, in patients under age 60, which factors are most predictive of severe outcomes? So it's a much more discovery oriented environment because we have all of the actual underlying data tables that come from those different institutions in those different models. And because of the hard work that you'll hear more about later that has been done on harmonizing those different clinical data models with one another, we can actually push all of those data in all those tables into one harmonized structure in the N3C cloud there, where this data can reside centrally in a secure enclave where we can perform machine learning and statistical algorithms and the like. So we can do things and we can operate in that context on those data collaboratively to build, test, and refine various kinds of classifiers and identify novel associations. And we anticipate that there's going to be some very interesting findings. We know nothing about this disease. It's obviously that's changing daily, but having access to the most robust data set that we could possibly have, we have opportunities to identify really interesting and important discoveries about COVID-19 and help our patients. Next slide. So this is just an overview of how the pipe works, the whole process. So we have our data partnership and governance. So really understanding legal and ethical responsibilities between ourselves as users, our institutions, and the NIH. You'll hear a lot more about that. We have the phenotype and data acquisition. There's been a large community effort led by Emily Pfaff at UNC to help the community define which patients should be included in the N3C cohort. And she'll talk more about that process. That data is then ingested and harmonized into the harmonized model that I talked about. And then we have our collaborative analytical platform, that secure enclave, where permitted users that have to sign a data use agreement with the NIH according to the use agreement, which is still under construction, but you'll hear more about that as well, can function together in a collaborative fashion where we can track contributions and results and share them rapidly. And the idea, of course, is that we do this very quickly, that we can actually reveal new discoveries and share them with each other and have scientific validation long before those scientific publications might come out, although that's, of course, also a goal. Next slide. So I'm going to hand this off to John now.
Hi, everybody. Can you hear me?
Yes.
Great. So first of all, thank you to Chris for that introduction. Thank you, Melissa, for your leadership in this project. It's been a real joy to be a part of this as it spins up. So I am leading the partnership and governance work stream for the N3C. And the goal here is to embed community-centered governance of the work. And so that is not just the governance work, which is I'm going to talk about, but all of the work streams you're going to hear about today. There's a commitment to transparency and engagement in all of these. I will tell you that you'll get as good as you give in engagement. And so to the extent that these things are interesting, sign up. There'll be links at the end where you can join and at least watch in on the various work streams, including governance. So what we do in this particular work stream is, as I said, work on community engagement. The vast majority of the work to date has been around drafting and feedback on key legal tools. So that includes the data transfer agreement from sites to the N3C cloud, as well as the data use agreement that moves the data back out for that use that Melissa just talked about. So we've done a lot of work. Christine Suver at Sage Bionetworks, my organization, has done a lot of the key work there. I wanted to recognize her work. So where we are now, and you'll see some information on both the data transfer and the data use agreements later in the deck, so I won't go into those. We've also been working a lot on single IRB and clinical protocol to enable this work. And I'll talk a bit about that on the next slide as well. As of this morning, we've started some work on standards and processes for how to attribute contributing institutions, mainly in the published literature that comes out of this, but in other areas as well. And then we also do work in supporting institutions during onboarding. So there's both the getting your data ready for onboarding, which is not what this group works on. We work on onboarding in terms of getting onto the common protocol. Melissa just alluded to the importance of the common data model and what it allows. We need to have common documents and governance agreements as well as we do that. And so we know that that takes some support, and so that's another part of what we do in this group. Our meetings, as I said, are publicly available and joinable, Tuesdays at 1 p.m. Eastern and Fridays at 10 a.m. Eastern. Next slide, please. So I'd like to thank Chris Shute and Johns Hopkins for volunteering to serve as the central single IRB for this project. The good news is that Smart IRB makes it easy. So all CTSAs are already members of the Smart IRB framework, which is more or less a treaty framework that is agreed in advance that makes it really easy to rely on a single IRB. So, so if you decide to be willing to use the single IRB approach, you do not have a lot of paperwork to do. It's a standardized reliance agreement under Smart IRB, plus an indemnification agreement with Hopkins as the SIRB. They, of course, already have 150 of those on file too. So the odds are that for the vast majority of CTSA sites, that most of the paperwork is pretty much done thanks to Smart IRB and pre-existing indemnification agreements. You do not have to do that. It is going to be more work on your end if you want to use your own local IRB. But in the spirit of flexibility, you can do that. And 2 sites indeed have already done that as well. So we do encourage everyone to take advantage of the single IRB under Smart IRB, but it is, of course, not required. And if you are interested in moving quickly on that just to get the process started, you can go ahead and contact Tricia Francis, who's been amazing at Hopkins, at the email address on the screen. Next slide, please. And this will be my last slide before I hand on to the next speaker, which is you're going to hear about the data transfer agreement, which is the ingestion into the GovCloud, N3C cloud. This is more about the data use agreement. So how do we pass the data on to researchers? So we're not going to allow download. And this means that we can have a pretty wide sharing regime because a lot of the most predictable risks and harms come from download and then secondary, tertiary transfer and so forth. The idea is to allow broad access, academic and commercial, to credentialed researchers. That's into the limited data set. That would be researchers from academic or commercial institutions who have committed human subjects training. And that's city training, I believe, is the standard that we're moving towards. And then also individual and citizen scientists who do not meet that bar of credentialing will be able to access synthetic data coming out of the system. And we will, of course, work on any other routes if it becomes necessary to create more styles here. But this is what we've come to so far. The idea is to keep this domestic, directed to COVID-related research. Since the researchers will be writing their code and their queries inside the enclave, those activities are recorded and can be audited in the event of a need to audit. We request disclosure of the results of your research back to the enclave for the public good, contributor attribution, which I mentioned is what the working group was working on this morning, and then an annual renewal as well as the no download. So, so this is the idea for how we provision access. It is essentially an objective review of the credentials of the data user, not a subjective review of that user's credentials or proposed research plan, other than to make sure it's connected to COVID. And the belief here is that this is the fastest way to get broad access to this data out in a way that we think is wise enough and wide enough to have the fastest possible impact of what we're doing, and as well as meeting the benefit risk calculations required of us as we think through the bioethics and the regulatory aspects of the data set. And I'll go ahead and hand off to the next speaker. Thank you.
Next we have Lily.
Hi, yeah, hi. Can you guys hear me?
Yep.
Yeah, we can.
Okay. Hi, this is Lily Portilla from NCATS. I'm the Director of Strategic Alliances, and my office has been assisting with the data transfer agreement. As John said, and Melissa had said, this is the agreement that helps bring data into the NCATS, into our cloud, and we have the data transfer agreement is actually available right now on our FAQs for this project. I think the link will be provided, and if not, I can put it in the chat box if you wanted to view it right now. But if you would like to request it along with some instructions on how to process the agreement, I have included here our email, and you can make that request. We can get as soon as we get your email, we will get the data transfer agreement to you. We are asking for contact information for the whoever will be deemed as the PI for the data transfer agreement, as well as who the authorized signatory will be for that particular institution. And this is information that we will need to be tracking. So far, we have sent all the data transfer agreement has been sent to all the CTSAs and a few non-CTSA institutions. To date, 6 have been executed thus far. As far as we can tell, the rate limiting factor right now is the IRB with some of the institutions that want to sign on. They're having conversations with their IRB, and that is what's delaying the final execution of the agreement. The comments that we have received on the data transfer agreement have been very positive. Because we are requesting no changes, we wanted to make sure that it wasn't too prescriptive. And the comments we've received thus far is that it's a very reasonable agreement. And as I said, if you want to look at it, please email us and we'll get it to you as soon as possible. Regarding the data use agreement, it is under review. We know that John Wilbanks and his group have done a tremendous amount of effort in putting it together. We are looking at it. I am a big believer of not reinventing the wheel, and we think it's going to be a great basis for the data use agreement. The only caveat or comment that I have is that, you know, being the federal government, there are some provisions that we are going to have to put in there that are going to make it acceptable for the government, since the government is, federal government is going to be a signatory on that data use agreement. So with that, I will stop and hand it over to the next speaker.
Good afternoon. This is Emily Paff, and I am with UNC Chapel Hill, and I am leading the phenotype and data acquisition work stream for N3C. And this is a work stream that's very technical and could very easily get in the weeds. For the purposes of this meeting, I'm going to be very brief in the interest of time, but I welcome questions in the chat and Q&A, and I will absolutely get back to you. So our work stream is dual purpose. They work together, but they are pretty different purposes. The first one, which is at least complete for now, is to work with the community to write and maintain a computable phenotype for COVID-19. And we have done that with some really amazing contribution from the community. We use GitHub very heavily as a sort of a home for all of our documentation and our phenotypes, and welcome to this day and in the future feedback from any community member on the phenotype or any of our code. So that's been going really, really well. And then the second purpose is to use that computable phenotype to define a cohort in a given site's common data model, and for us as a work stream to write and maintain a series of scripts that will execute our computable phenotype in each of 4 common data models: OMOP, I2B2ACT, PCORNet, and TriNetX. So the basic principle is that we want you, sites, to come with whatever data you have in one of those formats, and we will take care of the rest of the harmonization from there. Before we move forward, I want to acknowledge what we're calling the phenotype small group. We do have a large group once a week meeting, which I would encourage anyone on this call to join. It's a great way to figure out sort of what the updates are for the phenotype workstream. But daily we meet as a small group with the individuals that are listed here. And these are the folks that are working really, really hard to write this code and to make things as easy as possible for the sites. So thank you to them. Next slide. So our basic principle is to provide white glove service to the sites as you onboard. So we sort of see joining as a new site as a two parallel process. And the first row here you just heard about, where you've got to get all your regulatory ducks in a row, and then you're waiting for approval. In parallel, what we're hoping is that sites will begin to engage with our work stream, the technical team, so that even before you can transfer data, even before you've got your IRB approved, you can at least learn about how it will work to transfer data. And I'm going to describe that at a high level here. So what we're planning on doing is having multiple sort of office hour type calls per week, where sites can call in for a given data model and talk to the tech team about what it means to run our scripts, any questions they might have, any problems they're having, and we will provide immediate support for those sites to make sure that they can get up and running. Most of our interaction will be, if not over those Zoom calls, will be in our GitHub repository. That's where we're going to hope that sites not only get all of the code from, because it's all going to be there, well organized and well documented, but they're also going to be able to submit issues and other ways of getting in touch with us to let us know when there's a problem. Assuming that everything's going smoothly, what we then expect is that once appropriate technical personnel are assigned at each site, that person will be in charge of running the phenotyping code on a recurring basis, let's say weekly or twice a week, and getting data to the N3C repository. Next slide. So what it looks like to run our process locally is that we have this phenotype code, and we have a version of it for all of the data models that I mentioned, IGB2ACT, OMOP, PCORNet, and Trinetics. So whichever data model you're running, we have code for that. And it's also available for multiple database systems. So we have a version for Oracle, we have a version for SQL Server, etc. We're really hopeful that one of our scripts will be plug and play for you. And if it's not, we'll write one that's going to be plug and play for you. The next step will be to run a very lightweight local data quality check that essentially will only check for extreme showstopper issues, but it's really, really lightweight. We're not talking about running a full curation like you might do with your other data network participation. And then once that step is over, we have an extract code that will dump out all of the data into a series of flat files that can be exported either using our scripts or through your local processes, if that's easier for you. And then after that, you zip up the flat files and transmit them to N3C via SFTP. At that point, the data will be picked up by the data harmonization team, and that's what you'll hear about next. I mostly want to reiterate in my limited time here that support is available for all parts of this process, and that if we figure out that something isn't working in one of these 4 steps, we will fix it. So just remember that our entire philosophy here is to be a support for the sites and make your job as easy as possible. I'd encourage you to check out our GitHub if you haven't seen it. It's a link to the latest phenotype in the slides, as well as the extensive documentation that we've posted so far. And I will hand off to the next speaker.
Next slide, please. Thank you. And so you can see on the extreme left, I'm Chris Shute from Johns Hopkins, that zip file that Emily's team has just handed to us. And I'm deeply gratified by the cooperation that we've all had across many of our teams, and we couldn't have done our work without Emily's team's help. And further complement, as you see, the data models that would be reconstituted from the zip files we received, the Odyssey, PCORNet, ACT, and Trinetics. I also want to commend the enormous cooperation and collaboration we've had from those communities in terms of working out exactly what this means. So once we unpack those files, we're going to check the manifest. This all sounds like boring plumbing, and it is, but we want to do this at scale in a way that is careful and will ensure the most accurate mappings and decompressions as we're capable of doing. You can see that we're leveraging a custom variation on the Odyssey data quality dashboard in the middle. That's just a random graphic of what the dashboard would look like on some data, test data. And our goal is to ensure that we have conformance with the data model. And to the extent that we don't, we would either censor or flag that data and move it into the operating version. And well, of course, we'll return complete reports of data quality issues to the contributing sites with an hope that they can address them in future. Many of the data quality issues we will fix on our end. For example, we know that a lot of COVID lab tests will not have associated LOINC codes. We can do that in this harmonization process, but it will be a comprehensive data quality checks. And our data quality logic is really created as a hybrid across the methods used by all of the data quality communities. Even though we're using the Odyssey dashboard, it will be configured. It's a highly configurable dashboard to correspond to the hybrid checks that are relevant to N3C. Next slide, please. From then, we will take the instances and using a commercial tool, Adeptia, which is, oh, by the way, a workflow manager. So all our workflow on the previous slide would be managed in the Adeptia environment, which our team is coming up to speed with very quickly. As I said, we'll repair or encode aberrant data. We'll transform what we get in any data model that is given to us into the OMOP 5.3. And I want to, again, thank the CDM communities and the SAMVIT community for helping us generate these harmonization maps and validate those harmonization maps. It's a huge amount of work. It's a simple slide, but there's a ton of activity going on here, and leveraging those validated CDM to OMOP maps to create for each site a target OMOP instance as the canonical rendering for analytics. Next slide. Then finally, we're going to take the information across all of the contributed sites, merge them on a periodic basis into a versioned copy of the master OMOP instance for all of the sites. And we'll be able to pass that on to the Palantir analytic platform. Next slide. I think that's it for me.
Thanks, Chris. I'll take it from here. This is Justin Ganay from Sage Bionetworks, and I'll be describing the collaborative analytics work stream. As shown here, we are downstream of the harmonization work group and responsible for developing and maintaining a secure cloud-based analytical environment for the N3C research community. This environment will host the harmonized clinical data and provide a rich set of tools, allowing researchers to access, analyze data, and share results. To accomplish this, we formed 3 subgroups. As shown here, I'll provide just a brief description, but you'll hear more detail from the subgroup leads shortly. The portals and dashboards group is responsible for developing infrastructure and processes to enable access and exploration of the N3C platform. The tools and resources group is responsible for selecting and deploying third-party tools and data to enhance the N3C platform. Excuse me. And the clinical scenarios and data analytics group is responsible for selecting key clinical questions and developing analytic workflows and tutorials around these questions as a demonstration for other researchers. Next slide, please. So this slide shows a conceptual diagram of the N3C secure data enclave. As no data egress is permitted, all the tools for end-to-end analysis are required to exist within this secure environment. An overarching goal for our environment is to promote transparency and collaboration, and to enable sharing and reuse of research artifacts across different research studies. So the platform itself is the Palantir Foundry system deployed and hosted within the NCATS cloud environment. Among other things, Palantir was selected as it just completed a rigorous 2-year FedRAMP certification, which will allow us to host data with limited PHI. Over the last 2 weeks, in fact, we've had over 50 people from the community trained on this platform, myself included, and I must admit I'm quite impressed with the power of the system and how accessible it is to many different types of users, whether you're a power user or someone who's not analytically inclined. So starting from the bottom, this is to permit data analysis and researchers to develop and share purpose-built data sets for research studies, which will be derived from the master aggregated database that we received from the harmonization group. Above that, we have data modelers who will be able to develop analytical modules within notebooks using popular analytic languages like R and Python that can be chained together to enable more sophisticated workflows, which can also be shared with other researchers and studies. And finally, the research artifacts, visualizations, insights, results will be curated and shared within dashboards. And we will be building data and tool catalogs to enable reuse of these artifacts with other researchers who wish to leverage prior work. So next slide, please. Finally, we recognize there are many excellent tools and resources developed by the biomedical community. Shown here on the right, just as a few examples, our goal is to host many of these within the NCATS cloud and to allow them to interoperate with the Palantir environment. And an example just shown here is the Odyssey tool set, which provides excellent resources for data quality, cohort discovery, and analytics, and we want to make sure those are available to this research community. So with that, I'm going to next slide, and we'll pass it over to Joel Saltz.
Hi, I'm Joel Saltz from Stony Brook. And so I think one of the very exciting things about this work stream is this is already happening, the strong interaction between the different types of people who are needed to pull together a rapid and meaningful and impactful analysis of COVID-19. And so one of the things that people who have been at the front lines of the COVID-19 pandemic have discovered, and certainly we have, is that understanding the mapping between salient characteristics of COVID-19 and electronic healthcare data goes beyond what people who've been involved in data mapping over the many years really have looked at, because it is a new disease. So for instance, invasive mechanical ventilation is one of the key outcomes and things that occurs with this disease, and actually going through and obtaining really well-defined outcome information from electronic health care data on things along these lines requires some know-how. So this is something that I think our group, working with the harmonization group, will really be able to add very quickly, and the Palantir environment will make it possible to develop reusable tools and data sets that handle these sorts of things. Therapeutic anticoagulation is another area where really, at least we've had to work with the clinicians and the experts in this in order to reverse engineer these sorts of things. Characterization of laboratory data at various stages of diagnosis and treatment are also very important. So in addition to the ability for groups to discover new predictors, because nobody is obligated to use any of this work, we're really convinced that by combining clinical experts and people with expertise in tools and dashboards and informatics, we'll be able to really rapidly create building blocks that can be used in community COVID-19 studies. And actually, we've made a lot of progress even in the first few weeks of this in doing this. Next slide, please. So as I said, the key really is not just to share expertise, but to create reusable tools and libraries. And I've also gone through the Palantir training, and it is very impressive. And we really will be able to use this environment to offer reusable tools as well as modifiable tools that where the use of these things can be tracked and repeated. And again, information in the electronic health record is often really does require specialized insight that I think we'll be able to make available in a reproducible and reusable way. Also, characterizations of temporal change are going to be very important in this. Okay, so I'm going to hand it on to the next person. Thanks.
I am Andrew Williams from Tufts, and Chunlei Wu and I, who's from Scripps, are leading the subgroup on identifying tools and resources, and we're doing that in close collaboration with the leaders of the other two work streams or subgroups within this, and collectively we're to determine not only what is available, but how things are going to sit within the cloud structure and what's inside the Palantir cloud infrastructure platform within the cloud, and what sits outside it but still inside the larger cloud environment. Go on to the next slide. And we've done an initial scan of some of the packages and tools and commonly used analytic frameworks like Python, R, SQL, SAS, and Tableau that are can sit within the Palantir Foundry. And we're going to leverage some tools that are more mature and already ready to use in the specific way they need to be used for the common data model that all the data are being harmonized to. So the broad categories of tools that we're looking to integrate include the NCATS translator tools that have been developed, the Odyssey tool stack, as was mentioned. We saw a demonstration already of an excellent sort of CDM agnostic tool for cohort identification called LEAF, and a variety of uses of NLP. We can go on to the next slide. The translator work, so my colleague Chunlei Wu is deeply involved in this work, much more than I, and has developed one of the key components of it. But it comprises 3 different classes of tools that work together to mostly, up until now, I think, work at a level a little bit more basic science oriented than the clinical analyses that the clinical data we're contributing are designed for. So these classes are the knowledge providers, autonomous relay agents, and standards and reference implementations. So there's several different versions of these, and an ongoing process of developing different strategies for achieving what each of these classes of tools is meant to do and how they operate with each other. And Chunlei has already laid out a plan for an initial test of how some of these tools may work together. And again, there's an active process going on with Palantir to understand how they communicate with data inside the Foundry if they can't exist there. And Chunlei's laid out a process of evaluating what the requirements are for instantiating these tools in the environment. Go on to the next slide. So this is a little bit more about Translator, which, as is noted, is evolving. The kinds of translational knowledge bases involved there are described as including edges that exist between the variants and genes, variants, proteins, drugs, and so forth. So the analytics operate on those networks. And the classes of some of the tools that I think are involved in the initial implementation of this, BioThings, the Reasoner API, Smart API, and BioLink, you'll be able to use the links in this slide if you follow up to get to much more information about those. And there's a separate class without a link yet on inferencing and filtering tools. Go on to the next slide. There's a complete tool stack for analyzing data that are in the OMOP CDM that the Odyssey community has already developed. A lot of them are wrapped up into a single interface called Atlas, but some of them are not. The data quality dashboard has already been mentioned. There are other data characterization tools and tools for analyzing cohorts and cohort performance, as well as tools that accept cohorts that have been defined within the OMOP space and fed into pre-existing study pipelines for developing and validating patient-level prediction models or population-level estimations of exposure effects. And those will almost certainly all be supported. Go on to the next slide. There's a lot to cover in both the translator work and the Odyssey tools that we don't have time to cover here. So this slide deck will contain links that will allow you to learn more about how to use that. And I think before I go on, I'll say one of the, we've got excellent participation already. We're very pleased by the number of experts who are going to be assisting us in identifying these tools, evaluating how they can be implemented and whether they can be implemented. But we're going to be doing a survey as well to this broader community to help identify tools that are particularly useful to all the experts that are going to be leveraging this resource. On the next slide. I think that's it for me. Thanks.
This is Warren Kibbe at Duke University, and Dave Eichmann at Iowa and I are running this subgroup. It's portal and dashboards, and overall the objective is to develop really external facing entry point for research communities and help develop those internal dashboards for summarizing for researchers their resources and their activities. And a big part of what we're building out is really the attribution and recognition model to make sure that everyone who's contributing to N3C can be given credit for that, as well as the tools that people provide, other resources that they provide into N3C, and any other contributions they make. So a big part of the portal and the dashboards is really working out all the infrastructure necessary to make sure that happens and that we can move smoothly between all the organizations that are participating in N3C and the cloud environment that NCATS supports. So next slide. Oh, and I should say we have our open meeting that all are welcome to attend on Monday at 10 a.m. Eastern. So that's 7 a.m. for those of you on the East or on the West Coast. And the Zoom links there. And again, there's plenty of opportunity to participate with us. Next slide. So just to restate some of that, we're really supporting the public-facing entry point into the analytics environment that you've been hearing about. We want to make sure that we can support the requests and track all the approvals, so more of the administration side of things, as well as supporting the data dashboards that would go across the data builds, the different releases. Again, as Chris mentioned earlier, the Palantir environment allows very granular level of tracking and releasing of data. And we'll be building the dashboards necessary to expose all that. And then, of course, tracking the research that's going on inside of the environment, whether that's using the limited data set or using the synthetic data set, which you're going to hear about shortly. And of course, also tracking where all those folks are, the institutions. And again, part of this is making sure we can provide the necessary recognition and attribution to everyone. And then also the application dashboard, so making sure that people can see what models and algorithms and services are being attached in the environment. So again, if you'd like to get involved in this, please send either Dave or I an email and get involved. Next slide.
Yeah, thanks. This is Tel Bennett. I'm the informatics director at the Colorado Hub. I'm a practicing pediatric ICU physician, and it's my pleasure to be co-leading the clinical scenarios and analytics subgroup, where the mission is to identify clinical questions and in some cases answer them that will motivate identification and creation of COVID-19 focused data elements and core analytic workflows. We have open meetings, and the link there is in the slides on Mondays at 11 Eastern. Next slide, please. So among the tasks that we've identified with a number of clinician, contributors that we want to be able to do is to phenotype. So clinical phenotypes we plan to develop and validate include outcomes phenotypes, those that have good, medium, poor, for example. These phenotypes will be both knowledge-based or ontology-based and data-driven. And we also have a vision to develop tools to predict these phenotypes, tools that will ultimately be deployable as interventions in live electronic health records. The inputs to these phenotyping tools might be baseline and early encounter data, demographics and key features of past medical history, including cancer, hypertension, diabetes, that have been associated with outcome, as well as early laboratory results, vital signs, necessary support. There's a lot of interest in clinical symptoms and symptom history when available, as well as social determinants of health from those sites where that's available. Next slide, please. So, and there's some overlap between these two tasks, but we also have a vision to be able to answer questions that would fall under the umbrella of predictive analytics that might become early warning or clinical decision support tools. These frequently have multiple dynamic inputs, and these might be analytics for the need for organ system support. the need, for example, for non-invasive or invasive mechanical ventilation. So who's going to need to get intubated, who might not. Renal replacement therapy, which is becoming a constrained resource as well, and or extracorporeal life support in places where that's offered. There are interesting and important complications that have been identified. This is not a complete list, including venous thromboembolism and sudden cardiovascular collapse. And so, phenotyping and developing tools to predict those would be both interesting, important mechanistically, and potentially useful as interventions. There's also a lot of pathophysiology we don't understand. What is cytokine storm in these patients? What are the features and perhaps subphenotypes of ARDS, of acute respiratory distress syndrome, who is going to progress to acute kidney injury, and what is this recently reported pediatric inflammatory multisystem syndrome? So next slide. So this is my last slide, but the third kind of general framework that we wanted to build analytic resources that could support people answering include questions related to personalized treatment response. So methods to predict response to various combinations of the antiviral, anti-inflammatory, and other medications that are being tried in patients with COVID-19. We have begun to think about more complex but high value data types, not in the initial load of data, but how do we have a vision to get higher granularity signal data, images, text, and omics data from those sites where it might be able to come in to fuel other analyses. I want to mention that this subgroup is co-led by Peter Robinson at Jackson Labs, Heidi Spratt at UTMB in Galveston, with me, and we are establishing domain expert groups in acute and critical care, in nephrology, in pediatrics, in rural and disadvantaged populations, and I imagine there will be others in the future. So if there are clinicians at your sites that would like to be involved, please reach out. Thank you.
All right, so I think it's my turn, and luckily we're short on time, but I just have a very brief couple slides to update everyone on this synthetic data work. This is a relatively emergent part of the N3C program, and really has four big objectives. The first is really to establish a pipeline that will allow us to produce computation-derived synthetic clinical data, basically data that looks and feels like real clinical data, that we can do everything from statistical analyses to machine learning and sort of stops in between on, but that has minimal to no risk in terms of patient privacy and confidentiality, and therefore can be shared more broadly. And this is going to be built on top of a platform that we call the SHARE platform, which leverages technology developed by a company named MD Clone. And as this group spins up, we'll be sharing a lot more of our preliminary experience with that platform. Unlike other parts of N3C, this is still early stage, and so we really will be focusing on a pilot of this pipeline using data from ideally somewhere between 3 and no more than 5 pilot sites. And then we will be making this synthetic data available to the broader N3C community, and I think importantly, both via the cloud-based analytical tools and secure enclave that you've already heard discussed before, but also via a set of downloadable extracts. You can think sort of a model not dissimilar to the way in which the Mimic platform works. And our goal is to have minimum barriers other than registration as a researcher with some indication of the intended use of the data for people to access those types of resources. And then lastly, we are going to be doing a lot of work with the community to envision how we can expand participation in this platform over time, adding more data from the N3C data partners into the platform. So if we go to the next slide, this is sort of the schematic diagram. I often joke that if nothing else, informaticians produce diagrams of boxes and lines, and this is certainly an instance of that. And this is somewhat complicated, but meant to explain the interplay between the core N3C pipeline, in which partners are contributing data that is being represented in an OMOP repository and then made available via a secure cloud-based enclave, and then the MD Clone pipeline that will be piloting during this initial stage, in which we will ideally ingest data from the OMOP repository for the core model that we've already talked about, or if necessary, although less preferable, engage in direct ingestion of data from pilot data partners and load that into a cloud-based data lake that allows us to produce a longitudinal synthetic derivative on demand that, again, will be made available both within these cloud-based environments as well as downloadable extracts. Will point out, although I failed to fully edit my slides, that it's not solely just work from the team at WashU, but also critical leadership from a number of other partners, including University of Washington with Adam Wilcox taking the lead, as well as Regan Street Institute and Indiana University with Peter Emby, and our community co-lead, who's Atul Butte from UCSF. And we are in the process of spinning up our work stream, including our open community meetings, where we'll be able to get into a lot of the critical details that I'm sure you have questions about, and those will be happening next week. So I would encourage everyone to stay tuned for details on that meeting schedule and the opportunities to engage, both to sort of better understand what we're building, as well as to identify the opportunities to grow this over time.
Thanks, Phil. Okay, Anita.
Hello, this is Anita Walden, Assistant Director of CD2H. I'll briefly talk about the website and the welcome packet. Originally, when we placed the N3C project information on our COBOT website, it was really quick. We used an existing website. Today, we're launching a brand new website, or revamping the old website to show the N3C information. On this website, you'll be able to access general information about the effort, information about the groups you just heard present, how to participate, events. There's also a status update that we'll provide weekly that you can get either through the listserv, or you can come out here to the website to access it. And then when the data is available, you should be able to access the data through a secure portal through this website as well. We'll also place, among other things, we'll also have a welcome packet on the site. And so can you go to the next slide? And in this welcome packet, this is really to help those sites who want to share their data, is to help them get started. And in this packet that can be downloaded from the website is a PDF that's clickable, so hopefully it's easy to navigate. We have background information about the effort. There's information about the governance process and the IRB process, including a protocol. There's also a link to a survey, and in that survey, it's information we want you to fill out if you haven't yet that will help us help you. So please take time to fill that out. And also, most importantly, is contact information. We realize there may be a lot of information you heard today, and so we want to give you the opportunity to contact any of us if you have any questions or any suggestions at all. Next slide. And if those of you who want to participate on any of the efforts in designing this whole process, here's some information here for the different work groups and work streams. And things change rapidly, so we'll constantly update the website, and just let us know if you have any questions. And in the sake of time, next slide. We'll open it up to questions. Are there any questions we want to go over? Let's see. Okay.
We basically have 1 minute. There have been a lot of questions answered in the Q&A as well as in the Zoom chat. We will make your questions and the answers available on the website as FAQs. We also really, truly welcome everyone to join our Slack channels and continue to ask questions there. There's a lot of experts there to help answer questions and help design the system together. So we're really grateful for everyone's attendance today, and we look forward to seeing you on future N3C calls. And thanks very much to all of the speakers and to Dr. Austin for setting us off in the right direction, and for all of the partners that make this all possible. So thanks very much.